Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Azin Shahira binti Muhammad Azram, and I'm a biology student AS2016A. The title for my final year project is Morphological Studies on Selected Species of Araceae. The table of content for this final year project is introductions, literature review, problem statement, objective, methodology, result and discussions, conclusions, and reference. Firstly, introductions. The world's ecologies would be incomplete without plants. There have been a bunch of great investigations on the incredible variety of plants. There are two types of botanical research. One focuses on plants' external shapes and structure that known as phytomorphology, and the other examining plants' internal structure that known as phytoanatomy. Plants belonging to Araceae family has the most variable veins and architectures among angiosperms, yet they all share a basic agriculture features such as a hierarchy of veins order that create a network of interconnect veins. There are around 3,300 species in Araceae family, which include 104 genera, veins and herbs that may be found growing on or in the water make up the Araceae family of plants. There are several subfamilies within the Araceae, the most famous of which being the Lemaceae families. Families members may be found in the tropical and subtropical. Flowering plants such as Amorphophilus, Titanium, and Wolfia species have the biggest and the tiniest inflorescence, respectively, of any plants known as family. Okay, the next is a literature review. Scientific categorization is a system for classifying the living organisms. Kingdom, phylum, or divisions, class, order, family, genus, and species are the 7 divisions in this system. Above all, kingdom is the biggest and highest level of divisions, whereas species is the lower level of division that can produce fertile offspring and has 2 names in binomial nomenclature. Each of 125 genera that make up the Araceae subfamily has over 3,750 different species. Since there are so many different taxa, it is important to take note that each one has a distinct physical characteristic that set it apart from the others. The research looked at a number of other genera, including Philodendron, Alocasia, and Aprocina, before making their final conclusions. For each species studied and for physical features in leaf venations revealed throughout the inquiry, we provide this detail as well in this final year project. The problem statements. One of the most useful strategies for resolving taxonomy issues at the family, genus, and higher level of specificity is the morphological analysis of the leaf. Understanding how these plants are categorized in the Araceae family and based on their individuality so that they might be produced with new features that individuals who hybridize them want. The objectives of the study are: first, to investigate the morphological characteristic of leaf of selected species on Araceae. 2. To measure the leaf area on selected species of Araceae using image processing technique. And the third one is to construct a cluster analysis regarding leaf morphological characteristic of selected species of Araceae. Methodology. The 4 methods have been used in this project. First, sampling for length and width of leaf. There were 5 genera and 7 different species of Araceae that were researched in terms of leaf morphological features, and the leaf were collected and analyzed. As a quantitative measure of morphological features, 10 leaves was gathered from each of the species chosen for the study, and before the leaf was measured, they were picked in a variety of size. For smaller size, the ruler had been used to measure the length and width, and for the biggest one is a measuring tapes. Next is for arrangement, shapes, color, margin, and venation of the leaf. The leaf arrangement, shape, color, and margin were analyzed as a second technique for obtaining the qualitative data for each leaf. For the example, the leaf color, the score will be given from 1 to 3. 1 for green, 2 for golden green, and 3 for green with color bronze or veins. The next method is for leaf area measurement in image processing technique. In order to calibrate automate measurement, a line is drawn on the margin paper using a marker and a ruler. A 5 cm line is drawn on the margin paper side for calibrating reason, and the cell phone's camera was used to capture 3 leaves from each species, which are then saved to a computer. Image J leaf picture analysis. Surface area program is then used to prepare the image, and in order to quantify the leaf area, we use the image J to approach and extract the picture. The last one is construction of cluster analysis with past software. The qualitative data was transformed into qualitative data by using scale measurement in the past software. All data of the 7 selected species regarding the width and the length of the leaf will be taken 3 times to ensure the most accurate result possible. And the characteristics such as leaf color, leaf shape, leaf arrangement, leaf type, leaf venations, leaf apex, and leaf base should be turned into quantitative data to accumulate all the characteristics and into the software for the further analysis. Thus, the dendrogram regarding the selected species of Araceae can be finally be constructed. The result and discussions. For the result and discussion, 2 tables have been constructed and 1 dendrogram relationships between species of Araceae family also have been constructed. For the table 1, summary of leaf quantitative morphology on selected Araceae species. The quantitative morphology of the width, length, and the area of the leaf of selected Araceae species is summarized in table 1, and these measures are excluded from the taxonomic classification process that use hierarchical clustering, since the results will not be as precise given how widely these quantitative morphology characteristics vary for each species in each genus. The table 1 shows the qualitative morphology scale for each characteristic. The leaf will be scored from 1 to 3, depends on their morphological characteristic, and the score depends to differentiate the difference between of the leaf between each species. And the third one is figure 4.15, shows the dendrogram relationships between species of Araceae family. The 5 genus relationship of Callidium, Diffenbatia, Philodendron, Colocasia, and Epipremnium was qualified using average between group. The technique of data mining, known as cluster analysis, involved the divisions of the sample into group or cluster on the basis of information discovered within the data that describe the sample and their relationship to one another. The samples that are linked with the same cluster need to have a pattern of similarity among themselves, but they should be as distinct as possible to the sample that are connected with other clusters. This research explored the difference of morphological characteristics of the leaf of 7 different species from 5 different genera. Each of them has been given a name derived from the scientific word, making it easy to identify their label. A genus and the epithets are the 2 elements of an organism's name, and each has a generic name and their epithets. Conclusions. The study of leaf morphology allowed the classification of 7 different species according to their orders, genus, and species. The width, the length, and the surface area of the leaf are the example of the quantitative data, while the qualitative data contain information of the leaf's type, shapes, color, arrangement, texture, margin, and variations. In this study, the morphological characteristics of the leaf of selected Araceae species were effectively exposed. As a result of this research, it is easier to distinguish between the various Araceae species since it emphasizes the dynamic nature of many fundamental characteristics. Certain species may share the morphological trait with other species from a different genera, even though they do not belong to the same genus. This has been shown in a number of the studies. As for recommendations, due to the large number of different species that belong to the Araceae family, further research has to be conducted so that the additional morphological characteristics of these plants families may be discovered. Therefore, it is advised that future research investigate the native environment of these abundant species so that the knowledge regarding their characterizations may be made available to the generation that come after them. The last one is reference. This is the list of some of the reference that I use in these studies about the morphological studies on a selected species of Araceae. Thank you for watching.